Hello students, we are back for the study of classification of the fungi. As I was telling you, the class Ascomycetes will produce ascospores. Okay, in the previous lecture we have just started with that. The mycelium will be septate and branched. Asexual reproduction is by producing conidia, so conidia formation. So here you can see that the septate mycelium will give rise to a conidiophore, a vesicle, bottle-shaped sterigmata with chains of conidia by basipetal succession. These are also conidia formation only, but here you can see that the mycelium is going to give rise to a conidiophore which is branched, then the sterigmata is formed with a chain of conidia. This is a penicillium and this is the aspergillus. I've taught you about the sexual reproduction study in the previous slides. Please refer to them to understand. Coming to sexual reproduction in Ascomycetes, the name ascospores, ascus, whatever is produced by the sexual reproduction, and hence the name Ascomycetes. Okay, so if you go back here, this is the mycelium, okay, which is giving rise to the female sex organ and the male sex organ. Male sex organ is called antheridium, female sex organ is called ascogonium. Okay, in the previous one you found it as oogonium, that is in the lower fungi. In higher fungi it is called as ascogonium. Such a structure was not seen in the zygomycetes, if you recollect back. In zygomycetes there is no prominent sex organs present. Okay, but here you have a prominent sex organ present. They will come in gametangial contact. Okay, they come in contact. That time there are many antheridia developing. One of the antheridium will come in touch with the ascogonium. It triggers the ascogonium. Okay, so this is actually it will have a large number of nuclei. Okay, I've just shown one. This is called ooplasm, and the number of nuclei will be there. Okay, so as the antheridium touches the ascogonium, it triggers the ascogonium into a structure which is called as ascogonial helix. The term helix means it is a tubular structure. Okay, and you can find that this ascogonial helix will become septate cells, and this is called as a stalk. So to repeat again, as it is touching the ascogonium, triggers the ascogonium, and you can find that there's a stalk, and this is called the ascogonial helix. Actually, earlier they were not identified such a type of study. That is why aspergillus and penicillium, which are studying here, typical examples, were placed under a different class called deuteromycetes. Okay, so what you observe here when this happens, plasmogamy. Plasmogamy has already occurred, one of the first step in sexual reproduction. Okay, this ascogonial helix has got number of septate cells, and if you observe properly, every cell will be having 2 nuclei. Okay, so in the next slide you are seeing that this each such a this is one a typical example of a helix. Okay, part of the helix, where you can see that there are 2 nuclei in each. They are going to fuse, the haploid nuclei which are going to fuse to form a diploid nuclei. So every cell will have a diploid nucleus. Okay, and then you get a meiosis concept. Each cell of this will produce 4 nuclei, 4 haploid nuclei. Okay, and again undergoes mitosis. So the 4 haploid nuclei will become 8. If you observe, you can see that there's a fusion, karyogamy. First you saw plasmogamy, now you observe karyogamy, then meiosis, reduction division. The diploid became 4 haploid. Then again a mitosis. All the 4 haploid became 8 by mitotic division. Okay, so that's the concept. Okay, first you get the plasmogamy, karyogamy, meiosis, followed by mitosis, which is a part of the cycle. Okay, so every cell, what you're seeing, will become an ascus. Every cell of this. We call this particular cell, go back here, we call this as a dikaryotic cell. Can you guess what is the meaning of that? Dikaryotic cell? Dikaryotic means 2 nuclei are there in each cell. Okay, a dikaryote, this concept is called as dikaryotization. What is dikaryotization? Formation of ascogonial helix with each cell having 2 nuclei. Okay, so this is followed up here, and if you see this, each one will separate out to become an ascus. You can see 8 nuclei. 8 nuclei will get rounded off into ascospores. So inside this is the ascospores, 8 ascospores. Every ascus will have 8 ascospores in it. Okay, so if you understand this, this is called as the complete fruiting body, called as the ascocarp or the Cleistothecium. So what are these which you see? All these dikaryotic cells which were there, which have undergone these, have become ascus. So in plural form, asci. ASCUS is called ascus, singular. ASCI is asci. Okay, so many asci are formed, and all the sterile mycelium, whatever antheridium, mycelium structure, all of them will become protective for this. Okay, and you can see a hull layer getting developed at the top, at the surface. Okay, this particular structure is called the fruiting body, called ascocarp. So I think you can guess it out now. Ascocarp, asco word is there, no? Ascocarp structure is formed in sexual reproduction. Hence the name ascomycetes is given for this particular class. And you have already learned part of the type study now. When for example, if they're asking in the examination, explain the sexual reproduction in penicillium, explain the sexual reproduction in aspergillus, this is what you have to write. So what you have to write? You have to draw the mycelium with the types of sex organs. Then the antheridias are developing. One will come in contact with the ascogonium. Then there's formation of dikaryoticization, where a helix will have each cell with 2 nuclei. And all these concepts, there's a plasmogamy, karyogamy, meiosis, then mitosis, ascus formation. In each we'll have 8 ascospores, and this is what is the asexual. Sorry, this is called as a sexual fruiting body, called ascocarp. This ascocarp will fall in the soil and remains there only until there is favorable condition. And this, when there's a favorable condition, this breaks open, and all the ascus will rupture. The ascospores are released from this. Imagine 8 are there in each. All the ascospores will be released. Every ascospore will become a mycelium by germination, just like a seed germination. Okay? Yeah, I think you followed this. Coming to the next class. What is this class called? Basidiomycetes. Basidiomycetes will be having a mycelium which is branched and septate. This is the class which is having a proper septum formation. So you are seeing here, no, mycelium will be septate, or here, see, mycelium is septate. Each septum is called as dolichopore septum. Okay, so you can see that this is the parenthosome and the pore cap. They can ask for 2 marks. What is dolichopore septum? You should say dolichopore septum is the true septum present in the class Basidiomycetes. Earlier also you find it, but the septum is not so well developed in the lower fungi, that is in the Mastigomycota part of the study. Few of them will have a small septa, not well developed. That's why it's called pseudosepta. Here it's called eosepta, true septum, the proper septum. You can, what is this called? The formation of this structure is called as parenthosome. Okay, from the mycelium you get the parenthosome developed, and then covering that is the pore cap. This is a pore. Okay, this is a pore cap. Why this is so organized here is cytoplasmic movements occur very regulatedly. In the pseudo means false septum, nothing of this sort will be there. It will be just a cut off structure like this, and cytoplasm will flow simply. So there's no proper, the streaming movement, whatever you say. Okay, in cytoplasmic streaming movement. Here a regulated movement will be seen. That is the speciality of higher fungi, where Basidiomycetes will start showing this type of a septum. Okay, Agaricus, Puccinia, these are some examples for Basidiomycetes. Coming to the asexual reproduction in Basidiomycetes, they form 3 types of fruiting bodies. The mycelium can give rise to holobasidium. Means you find a structure, a vesicle called basidium, that will give rise to sterigmata with basidiospores. I want to tell you the remaining 2 here. The other one is called as a phragmobasidium. So you can see that the conidiophore will give rise to a split bottle-shaped sterigmata, which are very elongated, and tip will give you the basidiospores. And the other one is iridospores, where the teletospores, which is formed by sexual reproduction actually, those teletospores, one of the upper cells will develop a mycelium and give rise to basidiospores. Here the example for phragmobasidium is tremella, and here it is Puccinia. Okay, coming to the sexual reproduction part of the Basidiomycotina, here you don't find sex organs developed. 2 different mycelium, just like zygomycetes, no, they come closer and they start fusing. Okay, it's called as somatogamy. Okay, and this is becoming a diploid now. From there you get a basidium developed. You can see this, the nuclei will fuse and then they divide. You can find plasmagamy in the this is called plasmagamy. Then you find karyogamy. The diploid nucleus will become 4 haploid. Okay, and from this basidium you get the 4 sterigmata coming out. Okay, this is an extra one by mistake. So you have 4 sterigmata coming out, and every basidiospore will give rise, will germinate. During germination they show again meiosis, and then the septum developed. So sorry, mitosis. So the septum will develop and the nuclei will be distributed in each septal cell. This I have already explained to you. Okay, so we'll go to the fruiting bodies which are formed. Next class is the Deuteromycetes. Okay, Deuteromycetes is called the imperfect fungi. Okay, the imperfect fungi means the particular class doesn't have sexual reproduction. It lacks sexual reproduction. That's important to remember, because a 2 marks question can be asked, imperfect fungi. So please remember, or it's also called fungi imperfecti. Okay, because they lack sexual reproduction. All the fungi which is there in the environment which lacks a sexual reproduction is placed under Deuteromycetes. Actually, Aspergillus was also placed under the same, but later on they found the characteristics of plasmagamy, so they shifted it with ascus formations and all. So they shifted it to Ascomycetes class. Okay, so coming to this topic here, the mycelium is branched and septate like in the characteristic of a higher fungi. Okay, and of course doliprous septum, all those are similar. Coming to the asexual reproduction, there's formation of conidia. You can see the conidiophore is very short in the Deuteromycetes and giving rise to conidia which are different shapes. For example, if you see here, Fusarium will have a sickle-shaped conidia. Alternaria will have a conidia which is like in chains, but they have a longitudinal septum and transverse septum. These are characteristics to understand. Okay, but now I'll be telling you what actually they involve in formation of the asexual reproduction. One of them is called as the acervulus. Okay, acervulus means you can see that the mycelium will have a very thick structure which is very dense and dark in nature. Okay, so here you can find that the conidiophore is giving rise to conidia. Okay, which are just like any other fungus what you see. I just want to tell you the difference between the two. Okay, here if you see, generally when you talk about Deuteromycetes, the fungi will have very thick, very closer mycelium, packed mycelium. Sometimes they will be loose, sometimes they will be very tightly packed. This parallel mycelium, whatever is there, is called as fascicles. Okay, and the closely packed structure is called as cinematus. The fructification means the fruiting body structure is called as cinemata. Like how you say conidiomata here in the Ascomycetes and all, here you will call it as cinemata. Okay, so here Sporodochium is one of the types of structures where the mycelium is showing the same characteristic but loosely arranged, interwoven. Okay, so they give rise to conidiophores and give rise to conidia on the tip. The main difference is they have a cushion-shaped nature which is loosely interwoven mycelium. Okay, conidiophores will produce conidia. Example is Fusarium, what we learned in the beginning, sickle-shaped conidia. Okay, and I was telling you about the acervulus. See the difference? In acervulus, it is very thick, dense, and dark. Okay, conidiophores are septate, and it's giving rise to conidia. Example, Collodotrechum. Okay, and another structure is called as pycnidium. Pycnidium is a flask-shaped structure. Okay, where there is a opening called as ostiole. Inside this, you find the conidiophores developing conidia. Example is Diplodocea. Okay, and there are few other similar flask-shaped structures like Dendrophoma give rise to beak-shaped structure. Here is a almost closed ostiole. Here is fully closed beak-shaped structure. And this is Phoma, which is a small papillated structure. Okay, so you got the differences between the asexual reproduction. In case of Deuteromycetes, there is also flask-shaped structures. Please remember. That means imagine there is some fungus which is like Alternaria infecting the plants, leaves or vegetables. Okay, so that will show some typical structures like this. Sometimes you can, but mostly you can find the mycelial nature and study the conidiophores. Pycnidium and all it will be there. It will be there in the fruits or vegetables. It might get damaged while removing and observations and all. But you can observe them when you're in your practicals, when you're observing the spoiled vegetables and all, because they're mostly, all these are saprophytic, and few are parasitic also because they infect plants. Okay, so students, I've just given you the basic idea of all the different divisions. So what are the three important divisions? Gymnomycota, Masticomycota, and Amasticomycota. Okay, and most of our practical studies and the theoretical studies are related to higher fungi. We don't have type studies of lower fungi, but there can be a question, explain silent features of fungi means you have to explain about all these aspects, but individual type studies will be dealt in. So with all these basics, in the coming lecture classes, we will be learning about different form study. First, let us say Aspergillus. In that, what is the mycelial character? What is asexual reproduction methodology? And what is sexual reproduction methodology? What is the significance of the fungus? Okay, like that we'll be studying. So with all these lectures, whatever you have heard till now, please be alert and learn them properly so that type study becomes more easier for you when I'm using those particular terminologies. Okay, so all the best. Please go through, understand. If any doubts are there, please send it through WhatsApp or even any other mode. I'll try to answer them. Thank you.